Have you ever really looked at a flower and wondered how it actually works? Not just, its color or its scent, but the incredible biological engineering that's happening deep inside? Well, today we're going to unlock that secret. We're diving into the microscopic world of carpel development, which is the fascinating way a plant builds the very structures that will carry its legacy into the next generation. All right, so here's our game plan. We're going to start with the big picture, what I'm calling the flower's female fortress. Then we're going to zoom right in to see how its inner chambers are put together, piece by piece. From there, it's a deep dive into the journey to the egg cell itself, before we finally uncover the genetic and hormonal masterminds that are conducting this whole amazing show. So our journey kicks off with the pistil. I want you to think of it as the central fortress of the flower. It's a highly specialized structure, and it's designed with one purpose in mind: to protect the plant's most precious cargo, its future seeds. Okay, let's break down its architecture. Right up here at the very top, you've got the stigma, which is basically a perfect landing pad for capturing incoming pollen. Then, follow that down through this stalk, the style, which acts as a guide for the pollen tube. And that tube's final destination? It's this structure down at the base, the ovary. That's the secure chamber where the ovules, the future seeds, are kept safe. Now, a quick but really important clarification on some terms, because this is key to understanding how this thing is built. The fundamental building block is something called a carpel. Some flowers have a pistil that's made of just one single carpel. But in a lot of plants, including the one we're focusing on, Arabidopsis, the pistil is actually formed by several carpels fusing together into one single robust structure. So we've got our fortress. Now let's go inside and see how the inner chambers actually get built. We're heading right into the ovary to watch it get filled with those all-important ovules. Okay, so as we just said, in Arabidopsis, two carpels fuse together. But what's really cool is what happens on the inside when they do that. As they join, they form this central wall, which is called the septum. And what that does is divide the whole ovary into two distinct chambers, or locules, creating a really safe and organized space for the ovules to grow. And this is where it all starts. Right down at the microscopic level. We're looking at a cross-section of a really young ovary here. See those little bumps of tissue? Those are the ovule primordia. They're literally the very first buds that are going to grow into mature ovules, and they emerge from that tissue layer called the placenta. All right, now let's fast forward a little bit in time, and you can see everything's really starting to take shape. Those initial buds are growing, they're differentiating, and that central wall, the septum, is becoming way more defined. The internal architecture is clearly getting a lot more complex. And here we are, the final result of this stage. We can now clearly see these distinct ovules, and each one is connected to the ovary wall by a tiny little stalk called the funiculus. So the chambers are now filled, the structures are in place, and they're ready for the most critical phase of development. Okay, we've built the structure, we've filled it with ovules. Now we're going to zoom in even further, right into the heart of the ovule itself, to witness the incredible process of crafting the female gamete, the egg cell. So how in the world does a plant actually make an egg cell? Well, it's this beautifully efficient four-step process. First up, a special cell, the megaspore mother cell, goes through meiosis. That's the type of cell division that cuts the number of chromosomes in half. This makes 4 cells. Step 2, and this is where nature gets super efficient. 3 of those 4 cells just degenerate. They're gone, leaving only 1 survivor. Step 3, the nucleus of this lone survivor then divides 3 times, creating 1 large cell with 8 nuclei floating inside. And finally, step 4, cell walls form around these nuclei, and what you're left with is a tiny 7-celled structure called the embryo sac. And tucked inside that sac is the prize, 1 single egg cell, ready to be fertilized. This is just an absolutely amazing biological machine, isn't it? The precision, the timing, the structure, it's pretty breathtaking. But this doesn't happen by accident. It brings up a huge question. Who are the architects here? What is actually directing this microscopic construction project? Well, the architects, of course, are the genes. Their instructions are written right into the plant's DNA. So now we're going to shift from the what to the how by exploring this genetic blueprint and the master switches that choreograph every single step. So the key thing to get here is that this is a genetic hierarchy, a chain of command. Right at the top, you have a master regulator gene called a gamete, or AG for short. You can think of AG as the main project manager. It doesn't do all the work itself. Instead, it kicks off a cascade, activating a whole team of other genes. You can see some of them here. And they act like the site foreman, carrying out all the specific tasks of building the pistil. What's really fascinating is that when we zoom in on just the ovule itself, we see a totally different crew of genetic specialists at work. One group of genes, shown up here at the top, their job is to establish the ovule's identity, basically telling those cells, hey, your job is to become an ovule. Then a whole separate network of genes, which you can see in this lower pathway, takes over to construct its protective outer layers. It's this complex web of signals that ensures every single piece gets built exactly to spec. But here's the thing. A genetic blueprint isn't enough. You need a way to communicate the plans across the entire structure, in real time. That's the job of plant hormones. They act as the conductors, orchestrating the entire genetic symphony to create a masterpiece. So how does this orchestra work? It's all about a delicate hormonal balancing act, mainly between two key players, auxin and cytokinin. As you can see in the diagram, their different concentrations create a kind of chemical map across the developing pistil. High cytokinin in one area and high auxin in another tells a cell its precise location. It's this chemical gradient that patterns the entire structure, defining the top, the middle, and the bottom. So you see, it's not some simple top-down command. It's much more like a dynamic, constant chemical conversation. The genes, the hormones, and the physical tissues are all signaling back and forth, coordinating their actions to make sure that every single component forms in exactly the right place at exactly the right time. Which brings us to this really powerful concluding idea. The source material calls the ovule a complex assembly of tissues designed for one single purpose: successful reproduction. So the next time you see a flower, remember the incredible invisible story that's unfolding inside. A story of architecture, genetic programming, and chemical negotiation all working in perfect concert to build the future.